Hi, everyone. Welcome to Foresight's Molecular Machines group. Really, really happy to see so many of you here. And yeah, we've had quite the, I think, interesting year so far. We've had a really wonderful Designing Molecular Machines workshop. And yeah, had invited Alexis to that as well. And so I'm really happy that you're here now virtually to present your work. Thanks a lot. I do want to say that for the workshop, Aaron and I are currently working on the report. So many of you have asked for the videos for that, and they will, the report and videos will be published very soon for that as well. So just to give an update on this. And then one thing I want to quickly make people aware of is that if you're interested at the end of the year of meeting people in this group and in other groups, then please join us at Vision Weekend. That's in Europe and in the US. And that is basically our end of year event where we bring together kind of like the top contributors, fellows, mentors, and so forth in different fields, once in a castle outside of Paris, and then also in San Francisco at the Internet Archive, and a few private locations there. And so we have really wonderful speakers. We have lots of time for mentorship hours, and it's kind of like our end of the year celebration. So if you're interested in meeting people in this group and in other groups, and I know that many people here in this room are already coming, but I just want to make you guys aware that applications are open now. You can apply, especially if you're a member of this group, you can apply for subsidized attendance, which basically means strongly reduced to free ticket. And then there's like normal sponsorship tickets available for those of you who are more in the sponsoring position. Okay, well, that's all for me. I'm really, really happy to have Alexis here today. Thank you so much for joining. Yeah, we couldn't be more excited, I think, for your talk. Your name has fallen a lot in previous discussions and presentation already. So we're very happy that we have you here, actually, like, yeah, not really in person, but live with us to present. And without further ado, the stage is yours, and I'm going to share more info about you in the chat, but I'm sure that many people are well aware of your work. So thanks a lot for joining.
All right. Well, thanks so much, Alison, for the introduction, and thanks again for inviting me to speak. So it's super exciting. It's a super nice, interesting group of people. Yeah, so I think you all know what I'm going to talk about today, potentially. So yeah, I've been working for a few years now on the design of genetically encodable nanomachines, so nanomachines that have proteins, and we try to design those using computational methods. So try to design completely novel folds from scratch using computers, and folds that have some mechanical degrees of freedom. And so we try to design new proteins that can hopefully one day perform useful work at the nanoscale. So just one word, so I work at Institute for Protein Design at University of Washington in Seattle. And so we use these computational methods and develop new ones to design a lot of different proteins. And so I invite you to check out the papers if you're interested. Cool. So it's just a brief, history of what I mean by protein design. So I'm sure a lot of you know about protein folding. Since very recently, there was a lot of a new revolution in the revolution that happened about fold 2. But briefly, the idea is that you can represent proteins just inside computers, and then you can find the computational correspondence between a sequence and a fold. And so that's been very useful, to find new structures for given proteins of just known sequences. But then you can also revert the whole process by designing a fold, so specifying a specific shape that you want to target. And then you can use these algorithms to try to find a sequence that best encode that shape. And so traditionally, protein design has been mostly guided by finding, searching sequence space for specific folds by just minimizing energy of the sequence given the structure. And so we've been using Rosetta for that for a long time. And the idea here is that you can compute the energy of a specific sequence given a fold. And so that works pretty well, for the past decade, a lot of paper published, designing a lot of de novo folds. The problem is that often you end up finding, in this kind of energy space, you find very deep minimaps that correspond to the actual shape that you want to encode. But you often also have some off-target states. And this is what I'm showing you here. So more and more moving on to more probabilistic methods that use machine learning, where instead of just finding, the minimum energy for a sequence, we can just find the maximum probability of the structure that is encoded by a given sequence. And that's very useful because we can navigate the space, with much more efficiency And so we try to, and we tend to avoid this kind of like off-target states. So I'm going to talk about kind of like both approaches today, and mostly applies design of these nanomachines. So very briefly, so protein design, is useful for many things. And we've shown that you can use that to send proteins that bind to things, that catalyzes, that can make different shapes. For the past decades, we've published a lot of papers on that, so I'm not going to go into too much details. Just to say that we can make a variety of folds of symmetry, nanomaterials. We can make things that bind small molecules, that go into membranes, that process information. So it's really like a very fast evolving field, and it's very exciting. But so really what I wanted to do when I joined the IPD is not only to just make things that have folds, but really to try to recapitulate what we see in nature, that is this amazing, sophisticated machines that really support all what life does, right? So like processing information, processing energy, and so these things are very complex, right? Because they have very sophisticated conformational changes, and they process energy and information in very complex ways. And so the thing I wanted to try to do is really just start from first principle and build very simple machines that we understand, pieces by pieces, and then try to put them together to build more complex things. So of course, when we look at like what nature has evolved, it's incredibly complex and very elegant. But I think we can start by just redesigning like simple parts, simple machines, kind of like in the same way with macroscale engineering. So here I'm comparing like like the complex machines in nature with like this like 18th century poster with like a lot of different simple machines. And the idea is to start by designing very simple machines like that, and then put them together to try to create very complex protein assemblies, and maybe one day build things that are closer to the complexity of, for example, a phage, which is a very elegant and beautiful machine method signature. Okay, cool. So we published a paper recently that was kind of like the first proof of concept towards the design of such mechanical assemblies of proteins. And the idea here was just to start a simple concept of a machine, which is a two-component assembly, right, of an axle and a rotor, and try to implement that using proteins. So what we did is that we designed a library of machine components, essentially. So these axle-looking things and rotor-looking things. So we did that computationally, designing a lot of different protein folds. And then we designed an interface between protein components to direct assembly and solution, as well as to try to control the mechanics by controlling the energy of the interface between components. So this is what I'm showing you here. So this is a protein fold with like the axle in gray and the rotor in blue. And here you can see an energy landscape. So as you rotate the two components related to each other, you can see that the energy actually varies. And so this energy landscape we can control by designing the contacts of residues at the interface between components. So this is kind of a real proof of concept that we started with. So we went ahead and developed some computational methods to do that. So I'm showing you here an example of this. So again, we have designed this library of components that look like axles and rotors. And then we put them together computationally and model their mechanical degrees of freedom, right, by like translation and rotation. And then we design the interface between components by finding computationally the actual residue identities that would trigger self-assembly, as well as give this specific energy landscape shapes. So using this method, for example, I'm going to show you here an example of such mechanical assembly design. So this little rotary protein machine has two components, as you can see here. So it has a D8 symmetric axle. So it's an axle made of 16 copies of the same protein that would come together in solution to this high aspect ratio protein. And then we have two C4 symmetric rotors that are designed to come around it. And so this is what it looks like. This is the model. So we designed these things in the computer, and then as you can see here, this is size extrusion chromatography, just showing that we actually get assembly of the components in solution. If you look at the structure in more detail, you have this very interesting topology of interactions between components, where you can see that it kind of like looks like a kind of like cowheel topology. And so these interactions between components actually will constrain the mechanical properties. So if you do some like computational modelization of the mechanical properties, you can see that the two components can rotate relative to each other and are quite constrained in translation. And so if you look at the energy landscape, I'm showing here on the right, you can see that's very rugged. And so this, as you rotate, the energy varies, right? And so you have these like energy minima that are separated by 45 degree steps. So we're pretty happy to see that assembles a solution. And then we did some single molecule analysis using CRO-EM. And so we looked at these assemblies, frozen in ice, and looked at single particles and averaged millions of them. And we could find that we were able to actually resolve two different states in the particles. So here in blue and gray, you have the two different models that are just separated by a 45-degree rotational step. And here you have the electron density corresponding to these models, which we're able to find experimentally. And so this is a proof that the system can actually rotate according to this energy landscape into these two different states, right? So here you start at rotational states, and then as you jump to another energy well, then you find solid structure. And so this is what we were able to find experimentally. So we're very happy to see that the computational models actually were pretty consistent with data. So I'm not going to go into much detail here, but I can show you briefly that we designed now a library of these rotary machine components. And the idea was kind of like to try to explore the design landscape, as well as try to see, how good we were at putting some mechanical constraint rationally, as well as like crafting, sculpting very precisely the energy landscape. So here I'm showing you models of different symmetric rotary machines with like coordinate data that corresponds to it. And then we can see that, at least computationally, the energy landscape that we obtain are very, very different. And so we were able to like see with very high resolution the traditional characteristics of at least one designs. And now we're like further exploring these other designs using different methods. I'm sorry, I'm not sure what's going on. Oh, here we go. Cool. So we have designed this, like library of the machines, and so these are like really just like Fourier diffusive machines, right? So they are mechanically constrained to only rotate and translate a little bit. They do not have, they do not generate torque, right? They're just like diffusing in both directions. And so the thing that we try to do next is to actually create a motor, right? So once you have these like Fourier diffusive machines, in theory, you can add catalysis to them so that you actually bias rotation in one direction and generate torque in another scale. And so this is, very interesting because that's what nature has like leveraged to power a lot of these like nanoscale machines. And so that's like a very, very difficult challenge that we was to go after. So very briefly, the idea here is that to create a motor, what you can do is use Brownian what we call a Brownian ratchet mechanism. So the idea here is that, I'm sure you all know about that, but very briefly, the idea is that you have a system that can diffuse according to Brownian motion, and you can bias this diffusion by adding binding and catalysis to a small molecule, right? So here we have this rotary machine with like the axle in gray and the rotary in blue, and we design a site at the interface that can bind a small molecule and design catalysis on the small molecule. And the idea here is that you can then pretty much cycle between an energy landscape, which corresponds to just like the assembly in like a 3D Fourier diffusive states. And then you can jump to a higher energy minimum when you have to a higher energy landscape when you have binding of the small molecule. And because of catalysis, you just like cycle between these two landscapes. And so that, in theory, this is enough to power rotation in one direction. So we went ahead with that scheme and took some designs that we already had made that we knew had some like Fourier diffusive rotary mechanics. And then we redesigned the interface between axle and ring to be able to bind and do catalysis on these small molecule fuels. So we targeted a reaction that's like, energetic enough to, in theory, power rotation. And so we have access, interestingly, to that variety of different fuels, as well as to some molecules that can react covalently with binding sites. So that's interesting because that allows us to test how we can both like power directionality in rotation, as well as like completely stop rotation by adding this two-side inhibitor at the place. And so this is just like computational presentation of the energy landscape as you add molecules, right? So you can see that in gray, this is like the traditional diffusive Brownian landscape. And then when you add a small molecule, then you can see that you really change the energetic of the interface, which is what we're after. And then adding a small molecule inhibitor, you can actually introduce like huge energy spikes that in theory should be enough to stop rotation. We're able to show that we get actual enzymatic substrate conversion. You know, we have catalysis, and then we could show using other methods that this two-side inhibitor can actually react covalently. So we're pretty happy with that, and so that was, you know, a very interesting step in design. But the difficulty is really like to show, you know, at a single molecule level that we actually can modify the energetics and the mechanics of the systems, you know, in real time with like very high precision. And so we've been spending a lot of time and collaborations on using single molecule methods with like optical microscopy to try to track this molecule and this rotation properties, right? And try to see and prove that we can actually have directionality. So I'm not going to go much into details here, but we explored a lot of different approaches, either by fusing these proteins with DNA origamis or using very advanced optical microscopy techniques. I could show you briefly a few experiments that we're conducting right now that are, you know, still ongoing. So I don't have, you know, very definitive answers. But so what we've been trying to do here is like we actually look at single molecules, right, using optical microscopy, and then as we add the fuel or suicide emitter, we track the rotation of the single particles. So this is like what we would obtain. So so this is a plot showing displacement in X and Y of like a nanoparticle that's attached to the rotor. So you have a donut-shaped plot because the protein can diffuse, right? So you have this kind of like round shape. So we're able to observe, you know, free Brownian diffusion, which we're very happy with. And then we can add the fuel to the system and keep tracking the molecule. And you can see that we still obtain this kind of like donut rings, which is interesting. And after adding this suicide inhibitor, you can see that you completely modify the trend spike going from like a donut shape to a dot, which is, you know, indicative of like complete like stopping of the system by upon addition of the suicide inhibitor. So we're very happy with this kind of measurements, and so we're trying now to get a lot of data to try to really make sure that what we see is very accurate. We can track, you know, the number of rotation over time. And with enough data, we're able to like measure the actual torque that we can produce with the system. Cool. So you know, I obviously like so now that we know the goal of designing these digital machines, like we're trying to explore many different other opportunities to design more and more increasingly complex machines. And I'm just showing you a few here, and I would love to talk more about that if you're interested. But so, for example, we're trying now to design compound machines and gear-like structures, right? So these are systems that not only have one mechanical degree of freedom, but have multiple mechanical degrees of freedom, and sometimes even like coupled, so you can make things that look like gear, right? So this is very exciting for many reasons. You could also imagine designing things that can actually like, you know, walk along a track, for example. So this is like a little, you know, little like car-like design that can bind to a protein track and use rotation to like propel along the track. We are trying to send some light-powered motors. So the idea here is that you can bind a small molecule that can be light activable, and then use light to generate torque using 14 nanomachines. We can design things that instead of just rotating, can actually couple rotation with translation and have a system that looks like a linear actuator. So that again, like couple rotation with translation, and can be used to like direct motion of molecules along the track. A very interesting approach is also to try to replicate what nature has done with membrane-bound machines and try to harvest the gradients across the membrane to power rotation. And I'm going to talk briefly about that now. And then last, I'm going to talk about new approaches to design very much more complex machines, and these can rely on machine learning to go far beyond what they've been able to achieve so far. Cool. So nature has evolved these really elegant and amazing little nanomachines that I'm sure a lot of you know about. So an example of those are the ones that power the bacterial flagellum. And so these systems really just harvest the chemical gradients across the membrane, and by doing this simple mechanism can power motion of whole bacterial cells. And looking at these systems, by looking at the very recent protein structures that were published recently, you can realize that this actual mechanism is surprisingly elegant and beautiful. So you just have really a C5 symmetric protein that goes in the membrane with an axle that goes inside of it that's C2 symmetric. And then by simple protonation of the interface between the two components, this is enough to power rotation of the whole system. So this is really a very elegant and beautiful brain ratchet example in nature. And so we started looking at the system and trying to see if we could really capture the mechanisms in silico, try to show that not only you could capture it, but then also use it to design, rotary motors de novo. So this is just showing briefly that the protonation mechanisms by which this motor operates, we could really capture computationally by modeling the whole protein using Rosetta in silico. And we can see by modeling the energy landscape that the systems can jump between different energy minimum upon protonation. And this was very consistent to the actual mechanistic properties that we can that have been measured experimentally. Without going into many details, so we capture these mechanisms and try to see if we could design that de novo using computational protein design. So we came up with this kind of design, right? So these are completely de novo folds that can go in the membrane. So you have this rotary, this rotor part here in blue that self-inserts in the membrane, and then this axle in gray that goes inside the rotor. And then we design very precisely the interface between components with the same kind of principles that we saw in nature, with residues that can be protonated. And upon protonation, we actually disrupt this interface according to a specific energy landscape. So we went ahead and designed such things. I'm just going to show you now some preliminary data. So these are cryo structures of the transformative motors. Here's the model, again, with the rotor in blue and axle in gray. And this is the cryo structure at pretty low resolution. But you can already see that you have, all of the bars are here. You can see the rotor and the axle going through it. So we're pretty excited with that. And now we're trying to put this in vivo as well as in synthetic membranes. So we were able to express the systems in bacteria, and we can see some clear localization of the two components. And we can also purify away the membrane fraction containing the two components. And we're also trying to reconstitute the systems in synthetic liposomes. And so now we're moving on to trying to measure the rotation upon changing the chemical gradients. So this is also very experimentally technical, as some of you may know, but it's something we're pursuing right now. Cool. So I've shown you a few examples of what we've done so far with computational methods to design protein nanomachines. But of course, when you look at what nature has evolved, it's kind of humbling. So here I'm just showing you the bacterial flagellum. You can see the complexity of folds, symmetries, and assemblies. It's really beautiful. So we know that we need new methods, to be able to build bigger things, more complex things. And this is something that we've recently been looking into. And the approach we've been taking is trying to do what we call attenuation, right? So I'm sure you're all familiar with these kind of images, that are now, a couple of years old. So this idea here is that what was shown in 2015 is that you could use neural networks and then just invert them essentially and just ask them to produce things that they think make sense, right? So you get these kind of synthetic images by just asking the networks to refine and notice essentially. And so these obviously are not real, but they approach reality, and they can produce sometimes, very, very real images. And so we kind of wonder if we could use the same approaches with the newly trained neural networks that are incredibly efficient at folding sequences into actual protein folds, and try to use these networks to produce de novo folds, right? And so we have these two papers that came out recently that we invite you to check out, because it's like a real revolution in protein design. So the idea here again is like to use this like very efficient protein folding networks, and to try to sample them, to try to ask them to generate protein folds by adding some loss function constraints into the search, right? So I'm not going to go into much detail here, but the idea behind the algorithm I propose in these papers is that you can just start with a random sequence of protein, pass it to like a network, and then ask the network to fold that protein. And then we do that, and we look at the output of the network, and then we calculate some metric on these folds, right? By the way, we call loss functions. So they can be like, protein folding matrix. There can be some geometric loss functions. It can be anything you can specify in code. And then when you do that, you add mutations, and then you repeat that cycle by using, for example, Monte Carlo Markov chain sampling algorithms. And then after thousands of iterations, you end up with protein folds that actually look very, very good. So just an example here. So you start like a random sequence. This is what it looks like when you fold it. It looks like random garbage. And then you apply it. So you go around the cycle, and then you define the sequence, and then you can obtain these beautiful, well-folded symmetric structures. So here it's just by applying some symmetry class. So here we say, okay, we want just like a C12 symmetric protein. This is what we get. We can also apply some, more complex geometric constraints. So for example, here we want these two helices to have a specific angle, and then we can get this like very cool-looking corkscrew-like protein. So this is very powerful because it's completely, it's like it completely searched like the design space very efficiently, and generates, very, very diverse solutions for a specific design problem. And so that was, extremely interesting. So very briefly, I'm just showing here that like we try to really like sample the design landscape by using like genetic algorithm kind of thing. So here we can just like hallucinate just random proteins, and every time we find something that is good, we can select it and then let it evolve furthermore, and then we can just repeat these steps again and again and again, and generate like a very large diversity of very cool proteins that are at least computationally very fit. And so we did that in a couple of weeks, and I could generate like a very large library of different components, different shapes. And so that was really impressive, because traditionally this kind of design endeavor would take, months, if not years. And we're able to generate like a very large diversity of folds and structures in a matter of weeks. Just to show you here that, so we took a few of these hallucinated structures. So I'm showing you here a couple of models of like different symmetries and very cool angles. And we put them in the electron microscope to try to see if they actually worked. And we were very happy to see that, they folded as expected, and we could get some very cool, averages of the structures that corresponded to the actual designs, position designs states. Just to say that we kind of try to design things that are as complex as possible. So here, this design is a C42 Symmetry Protein, which is something that exceeds what we can actually find with PDB, which kind of shows that in theory we can design very complex things that that are far beyond what nature has samples in evolution. So we got some high-resolution structures, just showing here a few examples of those. Again, some like very high symmetry from C15 to C33 here. This is what they look like under the microscope. And we could get some high-resolution coherent structures with like 5-point harmony under like 2.5 angstroms for a few of those. And these things look absolutely beautiful and really cool. And again, these things would have been extremely hard, if not impossible, to design just a year ago. Interestingly, these folds are very different from what you can find in nature, which again is a strong indication that using these methods, we could design nanomachines that are increasingly complex and much different than what we can find in nature. Cool. So I think I'm probably going to stop here. There's many more things I wanted to show you, but I'm happy to answer questions. But just to finish, just want to say that, yeah, we're just like reaching this point where we can finally try to capture what, nature has evolved by using these computational methods. And hopefully we can start creating increasingly complex little machines, and maybe one day design things that are useful, like it's kind of like page-like machines. On that, there's many people I want to thank. Of course, David Baker, which is the director of the IPD, and then a lot of my colleagues at the IPD, my collaborators at the Coleman Lab that did REM, in the Bryant Lab at Stanford that did some psychomedical experiments, and a lot of other people. So thank you very much. And with that, I'd be very happy to answer any questions.
Wow, this was wonderful. Thank you so much. Very, very inspiring to see. And I think you already got a comment in the chat. Beautiful work. Yeah, well, thanks a lot for this walkthrough. We already have a question in the chat, from Ted, if you want to unmute yourself. I also want to say, if people would like to ask questions, feel free to drop it in the chat or raise your hand, depending on what you prefer. But Ted, if you want to have a go.
Yeah. Thanks for showing all these interesting machines. I think you partially answered my question later on in the presentation. I was mainly interested in how do you think this can scale up to put lots of these machines to get the work together in sort of more complicated machines. Sort of to what extent would you have control over, or methods to control individual machines within, say, a larger collection of machines? So there could be like focused light or some particular chemical that docks to some part of just one machine and not another, or there's some ideas along those lines.
Yeah, absolutely. I mean, it's a great idea. I mean, in theory, we can have very good control over either like assemblies and collections of machines or like single ones, right? So we could design different catalytic sites, different, we could either like just really design these things to be responsive to different molecules, right? And you could like have some kind of way to address them using light or the mechanisms. So yeah, I think like, collections of machines is really the next direction to go. To create, more complex mechanical assemblies.
I think that sounds like a really good direction. We have lots of people now have made individual machines in various ways, but one of the real difficulties from sort of an engineering perspective is, well, how do you put lots of them together?
Right, absolutely.
And have but still maintain some kind of control of them. So yeah, I think that sounds like a really good direction you're heading. Thank you.
Absolutely, thank you. I absolutely agree.
Wonderful. Again, in case anyone would like to ask a question, just raise your hand or drop it here in the chat. Again, the algorithm is I will ask the Foresight questions until other people interrupt me, which is the one that we always send out a question to speakers up front before the talk as well. But we're trying to also look a little bit like beyond the current horizon of where technologies are. And like for someone who's like new in the field, what would you like recommend that they potentially specialize in? Is there a specific challenge where you're like, okay, this would make my work much easier. Please, someone just tackle this. This is now your time to make requests.
Yeah, I mean, so in terms of like trying to push the next frontier of computational protein design, I think developing more efficient algorithms to do design is really what's really the limiting factor. And machine learning here has proven to be really game changing. So yeah, it's just finding new ways to apply machine learning to protein design is really going to change everything.
And do you have specific areas here? Like if someone is actually interested in taking this on, like do you think there's specifically promising avenues?
Mm-hmm. Yeah, I think so. So far, we've used these like machine methods so far to design just protein folds. It's only we only have the capacity right now to manipulate like natural residues because we've learned from PDB. the next frontier really would be to incorporate into these machine learning algorithms other atomic constraints. So, for example, the catalysis, right, or to do binding to like small molecules and things like that, to really try to bridge between sequence like protein folds and chemistry essentially. And so this is not really results.
And so would you like then say it makes a ton of sense for like people that are currently in machine learning to enter the field? Like, is that a specific, I guess, like, intersection that you want to further more? Because there are, every once in a while, we get emails from folks that are, have more of a computational background, but they don't really know exactly where to plug in. Like, are there any specific introductory resources or something that you think are really great?
Well, actually, I mean, it's such a fast evolving field, and there's so much stuff coming out right now. So every week, you have a new network that was trained on different things, and so it is hard to point to specific resources. I mean, it's very good to just start with like what DeepMind has published and what they that's very very good explanations of what they've done, and it's a very good way to start. And then just like following the literature on that. And I mean, it's really, right now, it's like really exploding, and there's so many different avenues that people are exploring. So it's kind of hard to point to a specific direction. But yeah, I think in general, just like, applying deep learning and new machine learning methods to protein design is extremely interesting.
Okay, cool. So more experimentation, I guess. And one thing I think that would be really interesting to hear is, do you have any and I know that it's a little bit more like kind of outside of the box, and folks don't like to speculate much, but if there are specific, kind of carrots that you think we could reach in the next 5 to 10 years, like if everything goes well and, progress continues the way that it does, like, are there specific applications further down the road that we could get excited about? Or like, what do you think is like, ahead potentially if we get things right?
Right. So I guess I just want to go back to what I was saying just before, what you were asking, because so as like these methods advance and become more and more powerful, it's interesting because like the limitation now goes back to actual experimental limitations, because now it's becoming very easy to design this like little machine things, but it's very hard to actually look at them experimentally at like the atomic scale. And so, for example, like program is like takes months before you get structure. If you come into the single molecule experiments, it takes like months or years. So I think it kind of also interestingly like put a lot of like power back in the hands of experimentalists in some ways. And so I think we should also really push, kind of like a whole area of like nanoscale, investigation of single particles with like time resolution. So yeah, this is very interesting, and I really encourage people to look into the biophysics of the system and try to develop methods to look into that. And then to answer your second question, so like, what can these things be useful for? Well, in theory, you can really, I mean, you can like, create nanomachines in the same way that nature has, has evolved these things to do, pretty much anything. And interestingly, so they can be genetically encodable, so you can like, put in cells, and you can like, imagine a lot of things. I'm showing this example of a phage because I think it's going to be the most like, intuitive one, is that if you could design a little, machine that can, scan cells and then detect, specific like pathological cells and then inject some information into them to modify them or cure them. This is really kind of like, a very simple case of a nanomachine that could be very useful, right? So
very cool. I like the pretty much anything approach. But all right, wonderful. I have now 2 other questions, and I think we'll take Ben first, and then maybe I'll ask Ted, and then Chris.
So I was actually going to ask this before. So this is kind of actually a follow on to your other question, which is like, do you have a sense of how we speed up the iteration cycles on like experimentally building these proteins? Like just sort of like obviously detail is better, but like how do we make that faster? Because it seems like that's like one of the cruxes, because we can't, like, it's unclear that the design tools are going to be able to like one shot a like function perfectly. So yeah.
Yeah, no, absolutely. It's a great question. Well, I mean, very often it's kind of technology. I mean, science is limited by technologies, and I think it's like one of these examples. Like, we need, more efficient microscopy techniques that are faster, cheaper, less footprints. And yeah, I mean, I don't have a good answer. But for example, like the cryo microscopes are constantly evolving and becoming easier to use and faster, and this is like helping a lot, So for example, we just got a new camera in the cryo microscope over the past month, and I just like that pretty much doubled the pace at which you could like collect data, and that has increase of throughput by a lot. So that's an example, like just improving like microscopic technologies, and then like for like the biophysical measurements of systems, I don't have a good answer. I think it's still very technical. But yeah, that would be it's a huge limitation.
So just to like make that explicit, the real bottleneck is sort of like knowing what's going on once you like knowing that you built the thing that you think that you built, that you wanted to build, and then knowing that it's doing, and then telling what that's doing are like the main bottlenecks to iteration speed.
Yeah, absolutely. So like, first of all, like getting a structure, so like Korean structures, it's, like takes like maybe a week or 2 weeks or 3 weeks. So that's great, like getting a structure, but then like getting like functional data, that might take months to years, really. And that's kind of like the bottleneck, I think, right now, unfortunately.
Awesome. Cool. I mean, like, that's not awesome, but it is awesome to have that made explicit. Thank you.
Okay, wonderful. Ted, I don't know if you want to ask a question or want me to ask it.
I'll ask it. So taking my second question there, have you thought about attaching several of these machines together in some kind of structure? And like, have you thought about design of the structure?
Mm-hmm. Yeah, absolutely. So this is something we're trying now. So I was showing briefly these, it's like a simple, like 2 components rotary machine essentially. And so it has two different axles that are inside the same structure. And so you have these 2 coupled wheels that can rotate. And so, it's kind of like a very simple version of like a little car, right? So that's like a very simple example of such machine. And so we're working on that. You could think about like more complex, things like that. Yeah, step by step.
Wonderful. Chris, you're next.
Oh, I think we can't hear you, Chris.
How's that? Can you hear me now?
Yep.
Okay. Could you speak a little bit more about how you designed the aldol hydrolysis catalyst within the context of the rotor? It seems to me to be a really, really sophisticated capability.
Yeah, absolutely. So, okay. So the first, so the reason why we targeted this like aldol reaction is because it is probably the one we can, design the best so far computationally. Catalysis is still a very, very hard problem, as you may know. And so this aldolase reaction is actually quite simple to design because it requires just a simple kind of like Tyrosine. So we know how to use like a few, like for example, a couple of lysines, like some backup, and it's like sort of well understood. And so we used like some computational methods that we have applied before to design aldolases in the lab that we know works. And so we just really took the pockets that exist between the axle and the rotor components and try to design in there a binding site for this aldol molecule, as well as like simple catalysis. And so for some of these designs, I think the simple catalytic site just has a lysine and then a tyrosine, and that's enough. Like placed in proper orientation, that's enough to get catalysis and turnover.
What's the role of the tyrosine there?
I am. So this is honestly not my specialty, but I'm not sure actually. I have to ask my colleague. So there's Yakov Kipnis here, which is like an expert on this kind of reaction and help me design these enzymes. Yeah.
Okay.
So I'm not sure actually.
Okay, and this is published already?
This is not published, so this is something we're still working on. But hopefully we're hoping to publish that, this year. But so like the work on the aldolase, reaction and computational design has been published like over and over. Yeah.
Yeah, I've seen that. Okay, thank you very much.
Yeah, thank you. Just to say that, like obviously, like it's a very, and that's why I was saying like we new methods to design catalysis, and so it's really like a huge area of research because it's still like resistant to like most computational methods, so it's definitely not solved. And this is a very simple reaction, but like if we wanted to create machines that have that generate like more power, then we might want to like target more different molecules, right? So right now, doing ATP hydrolysis, for example, it's really out of reach, unfortunately.
I guess one other question is, what's the turnover, like turnover rate?
Turnover rate, yeah. So it is around like 0.01 per second, which is pretty low. Yeah.
Okay.
Yeah.
Thank you.
Cool,
Yeah,
wonderful. I mean, feel free to, when you do publish relevant bids, just send them to me and I'll try to disseminate them in the group as necessary. But yeah, okay, cool. Again, if you'd like to ask a question, feel free to do so, raise your hand or put it in the chat. In the meantime, like one bit that, at Foresight we often push, and this is again more like a future-facing question, but is this notion of like differential technology development. Like I think, many of the technologies that I think are currently coming online, especially the ones that are machine learning like powered, are, have potential dual use possibilities as well. And I don't know, I just listened to this podcast recently on Lawfare about Fabio Urbina, Filippa Lenzo, Cedric Invernizzi, and Sean Atkins published this paper that I'll share here in the chat in Nature Machine Intelligence about dual use of AI-powered drug discovery. And I wonder if, that's something that you think about at all. Like, do you think at all about like infra hazards or, and like potential like misuse about these technologies? Or is that more something where like, no, we're just really trying to make progress for now and it's not really on the horizon yet? Because I think it's something where at least in the existential risk communities, people start caring a lot more about. And yeah, I'd love to see if you have any ideas. Like if anyone would have good ideas, I feel like it's you.
Sorry, the question was directed to me, or
yeah. I mean, like I think it's just I think really crazy how fantastic machine learning methods are for
right.
designs for all different types of molecules. And I wonder if you've thought at all about dual use, or like are there any risks that you already see like that are on the horizon where we're like at least like, hey, we should have an eye on this?
Yeah, I mean, I think yes, I thought about it, and it is so in terms of like machine design, I think very far from anything that could be like remotely dangerous. But like, so for example, you can use these algorithms to design things that look like toxins. And so I haven't shown you any examples of that, but we designed these like short peptides that can assemble in the membrane, for example, and these like resemble like natural toxins. So you could absolutely like it just like augments the possibilities in terms of design. So yes, you can imagine designing new toxins that have sequences that are far from what exists in nature, and so therefore it's very hard to control these things, right? Because so for example, now I'm sure like you're not allowed. Also, for example, if you wanted to buy a gene that encodes for a specific toxin, then you would not be able to, right? Because there's like you just blast the sequence and make sure that it's not something dangerous, or like viral sequences or bacterial sequences. But you could absolutely like go around that and redesign the toxin either by optimizing a natural sequence or like create one from scratch, and that would be extremely easy. And there would be nothing to prevent someone from synthesizing these genes. So yeah, this is a very interesting and scary prospect.
Yeah, I just shared the Nature article here, where basically I think those two researchers were invited to a national security conference on bio risk, and then just as a kind of like, you know, gimmick experiment, they're like, oh, what could we actually design? And they're like, oh, okay. And anyway, it was a pretty terrifying and like, you know, I think quite thought-provoking article. But okay, wonderful. Well, I think one thing I would also love to know, because I think that oftentimes we only hear about, like, I think research, you know, once it's already done. If you can share, I don't know if you can share any of it, but like, what is your guys' lab like going to focus on, like, in the next, you know, year or so? Do you have a plan for the next year or few years? Like, what are specific areas that you're hoping to address now?
Yeah, well, so I mean, many areas, I would say. So I mean, obviously, like, just, you know, trying to design increasingly complex folds is always, you know, a challenge. And then trying to move on to more functional systems. So, you know, so far, pretty much what the lab has published is things that fall in specific shapes and then bind other things, right? And so as we try to extend these capabilities, we also try to incorporate more functionality into these things. So, you know, some design new protein binders that can act as, like, you know, like some kind of like therapeutic molecules, right? So that, for example, trigger specific, like, you know, cascade reactions inside of cells when binding. So things like that. Trying to do more complex catalysis to you know, to create protein regions for the industry. One area that's very interesting and that I find, you know, very fascinating is trying to design protein nanomaterials. So that can be used, you know, for medicine or industry in many different areas. What else? I mean, you know, I think so. Yeah, I mean, we're exploring so many different areas. It's kind of hard to to remember sometimes. But
do you know, like, in medicine, is there a way to be more precise? Like, what exactly do you think could it be useful? Because we also have the biotech and longevity group at Foresight, and they are always asking me, so what's happening in the molecular machines group? You know, can we use any of the stuff soon? And right. So, you know, anything in particular that you think could be a cool application?
Yeah, so I mean, this whole field of, you know, designing protein binders that can be applied for a lot of different, you know, medical applications. One thing that's quite exciting right now is the field of, like, synthetic interleukins, right? So you have this company Interleukin that's been spun out of the IPD a few years ago, that have this kind of, like, you know, IL-2 mimetics, for example. So kind of like designing, you know, de novo folds that kind of can like, you know, restore some you know, interleukin based pathologies is one thing that's very interesting. And then, you know, we're developing binders for a lot of different pathologies. And this is not really my field, so I cannot comment too much about that. But yeah, this is a very vast, you know, area of research for protein design.
And do you have any ongoing collaborations with any companies, or like, are you guys advising any companies or something? Like, is there already like some kind of interlinkage happening, or?
Yes, absolutely. I mean, you know, as you know, there's like a lot of different spin-outs, or, you know, that are still, you know, part of the IPD and work with us, so that we're constantly, you know, interacting with them. Another field of research that's like, and translational research that I'm exploring right now is like applications for diagnostics, right? And that's quite exciting. So there's this company Mono that's gone out a year ago now, that's trying to apply this kind of like protein switches that are called Locker. I don't know if you heard of it. To do diagnostics, for diagnostics. And this is
what is it called? What is it called?
The company is called Mono, like this, and they have this very cool technology that's based on the protein system that is a switch, and so you can add, like, you know, like a little binding tool that targets a biomarker, and so as it binds to a biomarker, it exposes, you know, it creates like it restores like a luminescence module, and so you can actually measure luminescence, which is like gated by the binding of the biomarker essentially. And so they have this pretty cool technology, and so they're using a lot of, you know, other work from people in the lab, and it's quite exciting.
Oh, cool! It's the first time I hear about them. Yeah. Like, it's fairly fresh, right? That I think it says they closed 25 million seed financing in August. Crazy.
Yeah, they're doing pretty well. Yeah.
Okay. Wow, super cool. Okay, thanks a lot. Maybe like one thing that we always ask at the end of each session is like a shameless plug moment of like, if people now are extremely excited about your guys' work, what could they do to help it along? You know, I don't know if you are currently have open positions or if you're looking for funding for specific bids, basically anything that like immediately would advance your guys' work.
Right. So I would say funding for sure is always very useful. And then, collaborations are always, really sought after. So I was, mentioning like just the biophysics of single molecule, very advanced kind of experiments like that. It's really something that we always after. So more collaborators, the better. And of course, like, any kind of like smart algorithms to, further empower protein design is really would be super useful. So really, like, any kinds of useful collaborations are very, very welcome. And funding for these like many transformative projects that we are pursuing is always also most welcome. So
very cool. Thank you. I already saw that you have your email on your website, which I linked here and which we're going to have linked from the YouTube as well. Is there anything else I should have asked? Any final bits that you would like people to know?
Yeah, I'm not thinking of anything, but thank you very much. That's a very, very cool group, and I'm very happy to be part of that. It's been super interesting so far.
Yeah, thanks a lot for joining. I hope that next time we'll be in person again. We're already planning next year's workshop. And yeah, thank you so much for presenting. I think, I'm speaking in the name of everyone, that it's like very inspiring. It's coming out of your guys' lab, and it's a lot. It's a lot to process.
Right.
So thanks for giving us an update. I hope it wasn't the last time that we have you on. And this is just, I think, as you can see from the chat, people are very impressed by the talk. Thanks a lot. And it's just a reminder for all of you guys that we're currently planning next year's keynote seminar program. We already have a few lined up that are quite exciting, but if you want to nominate a future talk, I shared the form in the chat, but it's also always in our emails. It's a type form. It takes under 5 minutes to fill in. And that's a way to make sure that we kind of surface bids that you guys find exciting. I just shared it again in the chat. Thanks, everyone. Just a reminder, if you want to join Vision Weekend, there's a ton of subsidized kind of free tickets available for people in this group. That's definitely what the sponsorships are there for. So if you're interested in meeting people from this group and other groups, then maybe you join us in France or in the US. Thank you so much, Alexis. And I hope it wasn't the last time we speak. And I see all of you guys probably next month.
Thank you very much.
Bye, everyone.
Bye-bye. Bye.